This video will cover energy and enzymes in biological systems. In biology, we refer to free energy as the ability to do work within the cell. Since cells generally are considered to be the workhorses in biology, they're the entities that do all the work. But the problem is that cells can't make their own energy. In reality, nothing can make its own energy, but at least plants can harness energy from the sun. Animal cells, fungal cells, bacterial cells, they can't do that. They have to acquire their energy from an outside source. The term that we're going to use to describe this phenomenon is harness chemical energy. So if cells were able to harness chemical energy, more often than not, from the food that they eat or ingest, the food has a source of free energy that is going to be made available to do work. Well, in what form does this free energy going to enter cells as it is either eaten or ingested from the external environment? Well, it's going to enter cells in the form of chemical potential energy. So chemical potential energy is the matter that contains energy due to the arrangement of atoms or chemical bonds in a molecule. In other words, if we think of a typical molecule, of course, is going to consist of some number of atoms all linked together by some number of chemical bonds. The chemical potential energy is the energy stored in those chemical bonds. And if we can make that chemical energy available, that's energy that is going to be made available to do work within the cell. So in general, if we want to break chemical bonds, we're going to have to invest some amount of chemical energy to break those bonds. Think of a karate expert who's trying to break a board in half. Some amount of energy is going to have to be invested in order to break that board into two pieces. In contrast, if we form new chemical bonds, energy is going to be released. And this can be counterintuitive, because when we think of energy being released, we often think of the energy that's going to be released when chemical bonds break. But in reality, energy is going to be released when bonds form. And the reason why this is true is because if I'm taking two atoms and I'm trying to push them closer and closer and closer together, at some point the electrons of each of those atoms are going to come in close contact with one another. When that happens, the repulsion that exists between those atoms is going to cause those electrons to want to be further apart. But we're forcing them closer and closer to one another. So by releasing some of that stored energy, we're going to be able to relieve some of the stress, some of the repulsion, and allow the bond to form between the two atoms. I do want you to be aware that in biology, all systems are open, whether that system is a cell, a tissue, an organ, an organism, or even an ecosystem. The energy that's available to enter the ecosystem, is also going to be able to leave the ecosystem. This may not mean that the amount of energy is going to be equal. That's not always going to be the case. But certainly energy can move in and move out. So if molecules can move in, again, thinking about what we eat, if molecules can move in and there's some stored chemical potential energy available in the bonds that hold the atoms together in that molecule, then we would assume that it's also possible for energy to move out of that system, and that energy that leaves might actually be made available to do work within the system. Well, how do we get access to that energy? Well, in order to get access to the energy stored in chemical bonds, we have to allow molecules to react with one another. So chemical reactions are the processes in which physical and chemical properties of substances change as new substances with physical and chemical properties are formed. And oftentimes, during those chemical reactions, as new substances are going to be produced, existing bonds between atoms are broken, atoms are rearranged, new bonds are formed, and energy is going to either be released into the surroundings, or will actually enter the system from the surroundings. And that's all going to depend on the type of chemical reaction in question. In general, if we have a substance that enters into a reaction, we're going to classify them as reactants. If we have substances that leave chemical reactions or are generated at the end of a chemical reaction, these are going to be our products. Now in class, I'm going to go through several reactions with you to help you understand the difference between reactions and products, but for right now, this will have to suffice as the reactions that we will be looking at in more detail once we return to the classroom. So, as we're going to see in class, many of the reactions require some amount of energy to get started. And we can see that the amount of energy that's required to get a reaction started is known as the activation energy. And we can see that right here. So oftentimes the term EA is used as the abbreviation for activation energy. And as we can see here, the reactants are on the left-hand side of our graph. Notice that the axes are labeled here with the y-axis being free energy. This is the energy being available to do work within the cell. And down below is the progress of the reaction. This is just the time, how long the reaction has been allowed to progress. So here we have our reactants. okay? And here we have our products. You can see that AB and CD are going to AC and BD. So what allowed us to get there? Well, in order to get from AB to AC, clearly the bond between A and B had to break. Similarly, the bond between C and D had to break. In order for that bond to break, we have to invest some amount of energy, and that energy is known as the activation energy. We can see here that those bonds between A and B are starting to break. That's the wavy line. Similarly, this is the same case for C and D. That wavy line indicates that the bond is beginning to break. So that by the time we get to the products, A has formed a new bond with C, B has formed a new bond with D. But notice that these products are at a lower energy state than the reactants are. And what that means is that in the case of this reaction, some amount of energy was given off by the reaction. The products have less energy than the reactants, so energy must have been given off. Where did the energy go? Well, oftentimes it leaves the system, it enters the surroundings, and as it enters the surroundings, it's made available to do work within the cell. So while it's totally possible for energy to be released in a chemical reaction, it's also possible that energy is going to be invested or absorbed in the chemical reaction. So hopefully now that you've seen the reactions in class, or if you watch this video early, after you watch the reactions in class, you'll have a better sense of which reactions are capable of releasing energy and which reactions are capable of absorbing energy. In general, if a reaction has absorbed energy, the products have more energy than the original reactants did. So if all chemical reactions require some amount of energy to get started, thus the activation energy, where is the energy going to come from? Well, a lot of students often think that heat can be used as the source of the activation energy within chemical reactions. But the problem is, while this works when EA is really small, if we're dealing with a living system, heat energy invariably causes temperature changes. And organisms tend to live in very narrow temperature ranges. So if we were trying to use heat, say from the surroundings, as a means to overcome the activation energy barrier of our chemical reactions, we're going to increase the temperature of the organism. And that often causes cellular damage. So that's not favorable. We need another way to lower the activation energy for a chemical reaction. And luckily, there is a way that we can accomplish that. Accomplish that with enzymes. Enzymes are specialized protein molecules that lower the activation energy for a chemical reaction. So by doing this, by lowering the activation energy barrier, they allow reactions that would normally occur very slowly to occur much more quickly. So we say that enzymes act to speed up chemical reactions because they take reactions that would occur slowly and allow them to occur much more quickly by lowering the barrier that is necessary to get the reaction started. So we can see this with our energy diagrams. Here is a typical reaction, reactants over here on the left, products on the right. We can tell that this reaction has released energy because the products have less energy than the reactants do. But notice we have an activation energy barrier that had to be overcome before the reactants could be converted into products. What enzymes do is they act to lower that barrier. So here it's pretty obvious that the barrier in this reaction is lower than the barrier in this reaction. But notice that the reactants and the products are the same. So we're not changing the reaction. We're still going to generate the same types of molecules that we would through this chemical reaction. We're still going to use the same reactants that were necessary to get the reaction started. What's different is that the barrier to get that reaction started is much smaller. And of course, because that activation energy barrier is smaller, the reaction is considered to be catalyzed and can occur much faster than it would in the absence of that enzyme. So what are these enzymes and how do they work? Enzymes in general are protein molecules, although you'll learn later on that enzymes can also be RNA molecules, and they act to speed up chemical reactions. How do they do it? By lowering the activation energy barrier. Well, what is it about the enzyme that allows the activation energy barrier to be lowered? Well, all enzymes are going to have an active site. An active site can be thought of as a groove, a pocket, a cleft, a crevasse, or some part of the molecule that can bind to a substrate. And substrate is the biological term for reactant. And we've seen this in the case of hemoglobin. While hemoglobin isn't an enzyme, if you remember, the heme group sat within a pocket of the hemoglobin protein molecule. So if you think of the heme group as the substrate, or the reactant, in the case of the chemical reaction, think of that same process occurring with enzymes. While hemoglobin isn't an enzyme, proteins that do behave as enzymes will have some spot that allows them to bind to a molecule and get the reaction started. And you can see that down here. Here's glucose, here's the active site for this particular enzyme. This is hexokinase. And when glucose binds to the enzyme, something clearly happens. Well, what happens? The shape of the enzyme actually changes. By changing the shape of the enzyme, this is the step that actually allows for the reaction to have a lower energy of activation barrier. And while the chemistry is a little bit beyond the scope of your understanding at this point, what I can say is that the shape of this enzyme changing is known as the induced fit. So as substrate binds to the enzyme, the shape of the enzyme changes. The three-dimensional shape of the enzyme changes. And what this does is it actually begins the process of breaking those chemical bonds. In reality, if we think of the substrate molecule as being composed of several atoms linked together by chemical bonds, if we had some way to pull those atoms apart, so keeping in mind that if we had a covalent bond, by pulling those atoms apart, we decrease the ability of those electrons to be shared equally between the two atoms. So by pulling those atoms apart, we weaken the chemical bond. If we weaken the chemical bond, we make it easier for that bond to break. If the bond is easier to break, then the activation energy has been lowered, and by lowering the activation energy, we're likely to have that reaction occur much more quickly than we would in the absence of the enzyme. This is another view of how enzyme catalysis works. In this case, we have an enzyme known as sucrase. Here's the active site for sucrase. Here's the substrate for sucrase. This is sucrose. Sucrose binds to the enzyme. We don't clearly see the induced fit that occurs as a consequence of substrate binding to the enzyme, but here's the enzyme substrate complex. This is going to be a hydrolysis reaction, so here comes water. Water will then bind to the enzyme, again at the enzyme's active site, where sucrose is already bound, and upon binding, water is going to catalyze the breakage or the hydrolysis of the glycosidic bond between glucose and fructose, releasing the chemical products and allowing the enzyme to return back to its original shape. This is important because we're dealing with an enzyme catalytic cycle. After substrate binds and is converted into product, okay, the shape of the enzyme will return back to its original conformation and allow it to bind substrate again. So one more time, substrate binds to enzyme to form an enzyme substrate complex. Upon binding, the shape of the enzyme will change. When the shape of the enzyme changes, it facilitates the breaking of chemical bonds. That lowers the activation energy barrier. The reaction takes place, the products are released, and the enzyme can return back to its original conformation. Here's another view of an enzyme we saw in a previous slide, hexokinase. This picture looks a little bit more complicated, but in reality, it's not that bad at all. Here's the enzyme. Here's the active site. In this case, there are two substrates. This is a bimolecular reaction. Both substrates bind to the enzyme's active site to form an enzyme substrate complex. Upon binding, the substrates will be converted into products as the shape of the enzyme changes, facilitates the breaking of those chemical bonds, lowers the activation energy. At that point, the products are released, the shape of the enzyme returns to its original conformation, and the reaction can continue a second time. Well, this is all well and good, but as you saw in the activity from last week, it's possible for us to change the shape of a protein by changing the conditions that surround that enzyme. So we can change the pH, we can change the temperature, and when we do that, we often alter the shape of the enzyme by changing the chemistry that's necessary to hold the three-dimensional shape of the protein together. Well, now since we're dealing with enzymes, keep in mind that by changing the shape of the protein, we can also change the way that the substrate binds to the enzyme. So it's totally possible that if we change the conditions enough, we may actually alter the shape of the enzyme's active site, making it impossible for the substrate to bind to the enzyme and thus making it impossible for that substrate, i.e. the reactants, to be converted into products. If this happens, we have successfully denatured the protein. We've changed its three-dimensional shape, we've made it impossible for that particular protein to perform its job. In the case of a protein that behaves as an enzyme, this enzyme will not be able to catalyze the conversion of reactants into products. So what we know is that all enzymes have an optimal temperature and pH. They have conditions where they work the best. So in this example, we have temperature on the x-axis, and we have the rate of the enzyme catalyzed reaction on the y-axis. And you can see that there are two different enzymes, a human enzyme and a bacteria enzyme, that have two different optimal temperature values. In the case of human beings and most mammals, the optimum temperature is going to be somewhere around 37 degrees Celsius. That's just going to be true for most enzymes that are found in mammalian cells. But there are other organisms that have the capacity to function at much higher temperatures. So what that means is that even at those higher temperatures, the enzymes are not being denatured. The active site remains intact. The substrate combined with the enzyme form the enzyme-substrate complex, even at those elevated temperatures. The same can be said for pH values as well. While there is an optimal pH for all enzymes, in the case of pepsin, an enzyme that's found in your stomach that digests proteins, that optimal pH is around 2. But keep in mind that a different enzyme might have a totally different optimal pH. In the case of trypsin, which is found in your small intestine, the optimal pH is around 8. All right, last slide. So what happens when these enzymes are actually denatured? Whether we're changing temperature or we're changing pH or we're changing some other environmental condition, what we're actually doing is altering the bonds that hold together the three-dimensional structure of that protein. So at a particular pH value, at a particular temperature value, these bonds that you see here on the screen act to hold together the three-dimensional shape of the protein. But if I change the temperature or I change the pH, it's very likely that I'm going to disrupt one or more of these types of interactions. And when I do that, I alter the shape of the protein, and I make it harder for the substrate to bind. So denaturation often leads to an inability of the enzyme to catalyze a chemical reaction. We'll be exploring all of these topics later on over the course of the week and in much more detail as we begin our lab later this week, but I wanted this video to serve as an introduction to some of the more important topics regarding energy and enzymes. I hope that you found it helpful.